Okay, so today there aren't too many of us that people got caught by the snow. Also here, genetics was canceled, so I wonder if that has anything to do with this. But we're just continuing with our discussion of glomerular filtration. So exactly what's going on as we're first getting fluid into our nephron. Okay, so last time we talked about the fact that we have protein-free plasma, so the water in your plasma plus a bunch of little solutes, so glucose is going to move, salts are going to move, so sodium, chloride ions are moving into the nephron. So they're moving from this capillary bed called the glomerulus into Bowman's capsule, which is that sort of mouth thing that's catching the fluid. Okay. So they're crossing a barrier, which is made up of fenestrated capillaries. So we're thinking about the capillary endothelial cells, the basement membrane, and the epithelial cells, which in this case are called podocytes because they have these special foot processes and slit pores between them. So we have things crossing these cell layers, but we also have these big slit pores, these fenestrations. Fenestrations just means like a window that aid us in transporting the contents of the plasma into the nephron. So this is just a zoomed in picture, a histological side of what that actually looks like, showing us that compared to our little cartoon. So today we're going to talk about how we actually determine how much fluid is going to go into the nephron, so how much of your blood essentially the kidney is filtering. So hopefully this actually looks kind of familiar. Hopefully you're not getting flashbacks, but this is very similar to what we talked about when we were looking at capillaries in our systemic circuit in our cardiovascular system. So these are actually those same Starling forces. We're just now being very specific that we're talking about this specialized capillary, the glomerulus, and now we're talking about fluid moving into the nephron rather than fluid moving into tissue. But otherwise, these forces are essentially the same as when we were doing that filtration pressure. So we have our two hydrostatic pressures that we looked at before when we were looking at capillaries. We should remember that hydrostatic pressure, that's like a physical pressure from the amount of fluid. So in this case, the amount of blood in the glomerulus or the amount of filtrate in the nephron. So when we're talking about hydrostatic pressures, that's P, hydrostatic. So basically the pressure of the water pushing on the walls. So if we're thinking about the pressure of the glomerular capillary itself, we have the pressure of these water molecules pushing out on those walls and pushing into the nephron, into Bowman's capsule, into this space. So this direction is the direction of filtration. Filtration is the movement of fluid from the plasma in the glomerular capillaries into the nephron. So this is filtration whenever we're moving into the nephron from the glomerulus. Filtration. And I'm just going to draw our direction, right? Filtration is that direction on this image. So if we look at our other hydrostatic pressure, so again, just the pressure of fluid pushing on the walls. The other place we have fluid is in the nephron itself, in that filtrate, right? So this is where we're moving fluid to, but that means there is some fluid there. So our other hydrostatic pressure is in the opposite direction. So that fluid is pushing back towards the capillary, right? So this would oppose filtration. But we'll notice when we get to the actual number that your blood pressure, that pressure favoring filtration, that pressure of the blood in the glomerulus itself, is much, much, much higher than the hydrostatic pressure in Bowman's capsule pushing back. So that's also kind of a similar relationship to what we saw when we were talking about capillaries in the cardiovascular system. And some of our values in the tissue were essentially zero. So we'll see a kind of similar setup here. So that brings us to the pressure of our impermeable solutes. So oncotic pressure is essentially the pressure of proteins pulling water towards them. So we can think of oncotic pressure as a pull. Okay. So oncotic means proteins. I'll write down for our, let's say, pull on water. Okay. So we have two oncotic pressures that we're interested in. The first one on this list is the pressure of our proteins or impermeable solutes in Bowman's capsule itself. So they would exert a pull on water towards them, so to come kind of water them down. So that is our Bowman's capsule oncotic pressure. So that is the same direction as that hydrostatic pressure from the glomerular capillary. So we have both a push force for filtration and a pull force for filtration. The push is fluid trying to push out of the capillary. The pull is any proteins in the Bowman's capsule pulling water towards them. And then we have our proteins in the glomerulus. So glomerular capillary oncotic pressure. So that would be the existence of proteins still in the glomerulus. Okay. So they would tend to pull water towards them. So this would also oppose filtration. So those two go together. So we also have a push force, hydrostatic pressure, and a pull force, and oncotic pressure opposing filtration. So the push opposing filtration is any fluid in Bowman's capsule pushing back towards the capillary. And the pull is the proteins remaining in the glomerular capillaries pulling fluid towards them. So we can add on our numbers here. So we can immediately see that our strongest force is PGC. So that's this first force, the hydrostatic pressure of the glomerular capillary. 60 millimeters of mercury is our measurement there. So that's really strong. And that pressure is basically coming from your blood pressure, right? Because it has to do with the amount of blood that is flowing through this capillary. So we're going to see that one of the ways that we change how much fluid and other solutes are getting filtered out by the kidneys, one of the ways we change that is by changing how much blood goes into these capillaries. So that's why we can do it. This is essentially the number we'd be toggling if we were trying to increase the amount we're filtering or decrease. We'd change this hydrostatic pressure from the glomerular capillary, which is really essentially just the amount of blood going through this capillary. So our other force for filtration is Bowman's capsule oncotic pressure. You know, that's 0. This is because proteins don't fit through those slit pores and they don't go through the membranes in the capillary endothelial cells or in the podocytes and those fenestrated capillaries on Bowman's capsule. So none of the proteins are able to move into the nephron. So while they would theoretically exert a pull on water towards them, since they're not present in normal non-pathological conditions, we don't really have to worry about them if we're trying to do the math. So those are our forces for filtration, right? Because remember when we calculate a filtration pressure before we were doing net filtration, now we're just being specific, Same equation, just now we're specifying that it's glomerular filtration pressure. Okay, so we'll see this abbreviated GFP, and we'll talk about GFR, which is just the same thing but a rate. Okay. So this is just forces for filtration. Minus our forces opposing filtration. Okay. So of the forces opposing filtration, you can see that we have moved some fluid from the plasma into Bowman's capsule. So there is fluid in Bowman's capsule that does exert a hydrostatic pressure. So there is fluid to push against those walls. But it's much lower than the fluid in the capillary, which kind of makes sense, right? Because we were moving fluid from the plasma into the nephron. So it would be relatively hard to get a higher pressure in the nephron when the capillary is where fluid is coming in from in the first place. Okay, so that's about 15, the pressure of the Bowman's capsule pushing back towards the capillary. So that's that one. And the thing that's higher here, opposing filtration, is our glomerular capillary oncotic pressure. This is because we're leaving lots of proteins behind in the capillary. Right? We're filtering lots of water and the solutes dissolved in it into the capillary, but we're leaving behind all those proteins that we see in the blood. So they're kind of getting concentrated. So this is a pretty significant pull back towards the capillary for water. Yeah, because those proteins can't travel into the nephron, which means that they're staying in the bloodstream and they exert a pull on water. Questions about, no idea what that is. Questions about these forces, their directions. I know it might be kind of a surprise to see these forces again, although we have mentioned these various types of forces before. It was a while ago. Okay. Well, hopefully the second time around, the fact that we've seen these similar forces in the capillaries, adding them on, now that we're just talking about these same types of forces, but in a new area, so in the kidney now, hopefully that makes it a little easier to process than that first time around. Okay, but they're essentially the same. We just have different numbers, and now we're just specifying glomerular capillary and Bowman's capsule instead of just inside a generic capillary and inside a generic tissue. Otherwise, we have the same exact kind of situation as when we were doing that filtration pressure. So this is just a recap of what I was just mentioning. So we're separating out our forces that favor filtration. So favor movement of fluid into the nephron. So that's our hydrostatic pressure from the glomerulus. So the plasma pushing on those capillary walls and pushing into the nephron. So that's pretty high. And one of the things that makes this especially high has to do with the kind of microanatomy of this structure. So what we see coming in, this is our afferent arteriole. Afferent means 2. And then we go through this glomerular capillary bed, and then we come out the efferent arteriole. And one of the ways that you can tell these two apart has to do with their diameter. So remember that narrow little blood vessels are really narrow little any type of tube because of their smaller radius. They have higher resistance, so it's harder to get flow through them. So the fact that the efferent arteriole is narrower than the afferent arteriole basically means we have fluid buildup in the glomerulus. So it's moving kind of slow there. We have pressure building because it's harder to get blood out of the glomerulus than it is to pump blood into the glomerulus due to those differences in radii and therefore differences in resistance. So that's part of why this number 60 is so high. Our other theoretical pressure for filtration would be the Bowman's capsule oncotic pressure, but we shouldn't have proteins in our filtrate, so this should be 0-ish. Opposing filtration, we do have some fluid in Bowman's capsule. This is because we have moved some water into the capsule. So if we were comparing it to the number in the systemic capillaries, it is a bit higher, but it's not nearly as high as the pressure in the glomerular capillaries. And then our oncotic pressure in the glomerulus is pretty high because we have a small volume of plasma because we've been removing its water. And so we're concentrating these plasma proteins. It's just another way of thinking about the same sort of thing. And a zoomed in picture for your studying purposes later. So we can see here we've labeled the afferent arterial, the efferent arterial, and all those forces that we're interested in, so those 4 starting forces. You can see that we come out with a glomerular filtration pressure of about 16, which you don't have to memorize this number, but we're gonna keep it in mind as we think about comparing this to what we had going on in our systemic capillaries. So now we're thinking about the glomerular filtration rate. I'm sorry, this is another case where we're going to have a bunch of pretty similar terms as we're talking about the glomerulus. So our rate, our glomerular filtration rate, that means we're thinking about like an amount per minute, whereas our pressure, we had an actual pressure like millimeters of mercury. So that's the difference here, but they're going to be inextricably linked, basically, because it is the glomerular filtration pressure that is causing us to move fluid, which is why we have a rate in the first place, right? If there was no pressure, we would have no fluid movement and therefore no glomerular filtration rate. So our glomerular filtration rate at this normal glomerular filtration pressure of 16 millimeters of mercury is about 125 milliliters per minute. Okay, and this is a number, a baseline number that you should be familiar with. So this 125 milliliters per minute gives us 180 liters of fluid filtered per day, which we talked about last time. That's a ton, right? Which is why it's important that we are able to reabsorb water so that we can maintain blood volume. It's not possible to lose 180 liters of water per day. You do not have that much in your body in the first place. So yeah. The renal plasma flow is slightly different from this glomerular filtration rate. That's the amount of blood that is just passing by, like passing through the glomerulus per minute. So we're not removing all the water. We're not filtering out all the water that was ever in the plasma. We're not leaving ourselves in the efferent arterial with just like dry little granules of pleurothene, right? So some of the plasma is still passing by and ending up in your efferent arterial and eventually in your veins, right? So we can note that this glomerular filtration rate is a lot higher than our filtration rate was for our systemic capillaries when we were talking about net filtration pressure. When we were talking about that relationship between capillaries and our normal systemic tissues, we had a filtration rate of about 3 liters per day, which was much more reasonable. So we had that filtered, then returned largely by our lymphatic system. But now with our GFR, it's much higher. And this also reflects a difference in our filtration pressure. So when we calculated out net filtration pressure, a normal-ish filtration pressure in the systemic capillaries is about 2 millimeters of mercury, which is way lower than the 16 millimeters of mercury that we have in the kidneys. Okay. So now we have another number. So far we can make kind of like a checklist, right? So we've had glomerular filtration pressure, we've had our glomerular filtration rate. It's a little clearer. And now we're going to talk about the filtration fraction. Okay. So what this is telling us is basically like essentially what percent of the plasma passing through the glomerulus is getting filtered into the nephron. So this is just helping us compare how much of that fluid is moving into our kidneys versus how much is passing it by. So all we need to do for the filtration fraction is compare the amount of fluid that got filtered into the nephron. So essentially that's our GFR. And we're going to divide it by our renal plasma flow, which was the total amount of blood that went through the glomerulus ever. So this is really a fraction, which is the same thing essentially as a percent. So we have our 125 milliliters of fluid that do end up going into the nephron. We're dividing it by the 625 milliliters of plasma that passed by in the glomerulus in total in any given minute. So a normal filtration fraction is about 0.2 or 20%. That's like the normal amount of the fluid that you remove from the plasma as you go through the kidneys. About 20% of the blood that you route through your kidneys ends up, at least in the beginning of the nephron, before we go through our water reabsorption. Which brings us to filtered load. I should have left those. So we've done our pressure, our glomerular filtration pressure, which had to do with the Starling's forces and how much force is there for fluid to move. Then we did our glomerular filtration rate, which is just how much fluid are we moving per minute. Then we did our filtration fraction, which is what percent of the fluid that goes to the kidneys is getting filtered into the nephron. And now we're talking about filtered load. So filtered load, we're no longer thinking about the fluid itself. Now we're thinking about like the stuff dissolved in the plasma. So usually really we're thinking about glucose, just very relevant clinically, like in your diabetic patients potentially. So all we're doing here is multiplying our glomerular filtration rate. So that was the amount of the fluid that was getting filtered into the nephron. So that was our 125 milliliters, which is 20% of that total blood flow. Just a minute. And we're multiplying it by the concentration of that solute in the blood. So basically we're saying that, right, like we're moving 20% of the plasma into the nephron in any given minute. So essentially we're also going to be moving 20% of the glucose along with it. So we're just multiplying the amount of fluid times the concentration of that molecule in the fluid. We only do this for molecules that are freely filterable. So all that means is like molecules that can cross that barrier of the endothelial and epithelial cells between the glomerulus and the capillary. So like our proteins that stay in the capillary that are too big to get into the nephron, we wouldn't do this calculation with those proteins because they can't get across that barrier. So usually thinking about glucose, because glucose is freely filterable, it's small enough and it is able to pass through those fenestrated capillaries. So our filtered load is going to be a quantity, so an amount, because if you multiply, basically our filtered load is an amount. So it's going to depend on how much fluid is passing plus that solute. So we could do our multiplication out. So here's our GFR. So this is our rate of 125 milliliters per minute. The amount of glucose we have in every milliliter, right? So in every milliliter is 1 milligram. So if we multiply those together, we can see that we have 125 milligrams of glucose per minute being filtered into our nephron. So that would be our filtered load, 125 milligrams per minute. I wouldn't worry about this calculation with the plasma glucose, but if you flashback to the physiology lab, good, it should. I think you did this calculation before. Maybe not. So now we're going to talk about the regulation of this filtration rate. So we know we're filtering 180 liters of fluid per day. Right? So 180 liters are coming out of your plasma and going into the nephron. We also know that we are not peeing out 180 liters of fluid. We're probably peeing out like 1.5 liters of fluid per day. So 1.5 liters of urine getting excreted. So 99% of that fluid, that filtered fluid, is getting reabsorbed. And we looked in our last packet, or first packet, at some of the ways we control water reabsorption. Right? So we were looking at our antidiuretic hormone and how that controls reabsorption in the distal tubule and in the collecting duct. We also looked at the proximal tubule and how we have a bunch of bulk reabsorption of water in the proximal tubule following our ions that are getting pulled back out of the filtrate and into the plasma. So we had reabsorption in both the beginning, the proximal tubule, uncontrolled, and then in the distal tubule and in the collecting duct, we had this controlled reabsorption of water, which is mediated, which is controlled by antidiuretic hormone. So we already know a fair bit about why we are not urinating out 180 liters of fluid, right? We've talked about some of that reabsorption. So this is basically explaining the second part. It's explaining that one slide we skipped from the balance packet. So we were talking about the end, like how we can regulate how much water is being reabsorbed. But another thing that we want to pay attention to when we're thinking about amounts of urination and amounts of fluid excreted is the changes to the glomerular filtration rate. So changes to the amount of fluid that is going into the nephron in the first place is going to change how much fluid you excrete, right? So like if you increase glomerular filtration pressure and increase glomerular filtration rate, so you're increasing the amount of water essentially going into the nephron, then you're probably also going to be increasing the amount of water you're peeing out, right? So if you increase the rate of fluid entering the nephron, you're probably also increasing the rate of fluid exiting the nephron. So we're also going to see that regulating that rate is an important way we can control how much fluid we're using to urination. So in addition to regulating the reabsorption of water, which is what we talked about first, we're also regulating the filtration rate, the glomerular filtration rate, because that's like our input of water to the kidney. So we're regulating reabsorption, how much goes back into the blood, but we're also going to regulate how much water goes into the nephron in the first place. So this is a graph of how the glomerular filtration rate, so that's our GFR, the amount of fluid going into the nephron per minute, changes with respect to our mean arterial pressure, which you remember is basically a sort of sideways measurement of blood pressure, right? So we can see overall, as our mean arterial pressure goes up, right, as we increase our blood pressure, we are going to increase glomerular filtration rate. So if you have a higher blood pressure, that means you have more force, more hydrostatic pressure of fluid in those glomerular capillaries pushing into the nephron, right? So that ultimately does increase your glomerular filtration rate because they're increasing the pressure. But we also have this plateau in the middle, right? We don't just have a straight line going up one-to-one. And this is because we have a bunch of regulatory mechanisms to kind of control that rate within a pretty normal range for mean arterial pressure, right? Not everybody has the exact same blood pressure. Not everybody is a textbook. So there is a rate, like there is a pretty wide range of mean arterial pressure where we can control that glomerular filtration rate and keep it steady. So it's only at the ends, really. So when we have way low mean arterial pressure or way high mean arterial pressure, that we see big changes in the glomerular filtration rate. Okay. So now we get to the reason one of the things you were labeling for those anatomy worksheets was the juxtaglomerular complex, because these different types of cells neighboring the arterioles going into the glomerulus and out of the glomerulus, and also neighboring that ascending part of the loop of Henle, the distal tubule. These are going to help us control that glomerular filtration rate by controlling essentially what's going on with the blood coming into the glomerulus. Okay, so we have three ways that we can control the glomerular filtration rate intrinsically. So when we say intrinsic regulation, we're just talking about really local factors, right? So we're thinking about cells in this region rather than thinking about like neurons and things from the brain coming in. So whenever we talk about intrinsic regulation, that's what we're talking about, just super local nearby factors. Okay. So our first way we can control the glomerular filtration rate is by controlling the smooth muscle in the wall. So myogenic regulation, whenever we see myo, we're thinking muscle, right? So myogenic regulation has to do with the smooth muscle in the walls of the arteriole, right? So here we can see those granular cells, or I believe that we also can call them juxtaglomerular cells, right? You see them surrounding the afferent arteriole. So they're surrounding this blood vessel going into the glomerulus. So they're surrounding this blood vessel going into the glomerulus. So if we have an increase in blood pressure, we have an increase in that, this is going to stretch that blood vessel just like it stretches all of our other arteries, right? We talked about stretch as we have blood pumping into the aorta, for example. So we know that when we have increases in the amount of blood, increases in blood pressure, this stretches the walls of blood vessels. So in myogenic regulation in the kidney here, those granular cells, those smooth muscle cells around the afferent arteriole, they sense that stretch and they snap back. So they contract in response to stretch. And that's all that myogenic regulation is. So as we have those walls stretching in the afferent arteriole, we are increasing the radius, therefore decreasing resistance, ultimately meaning that we had more fluid going into the glomerulus and therefore going up through the kidney. So this myogenic regulation, because we're now snapping back in response to the stretch, we're contracting in response to the stretch, that's decreasing our radius of that blood vessel again. So it's decreasing the flow into the glomerulus. So when we make that blood vessel smaller, we make it so that there's less blood going to the glomerulus. So we could add on this contraction, decreases GFR. our second method of intrinsically regulating our glomerular filtration rate, something called tubuloglomerular feedback. So this should tell you that it has something to do with the tubes. So here we mean essentially the ascending limb of the loop of Henle or the distal tubule. So that basically the end part of our nephron neighbors the beginning part of our nephron, right? We loop back around, so the end and the beginning are kind of touching. So we can see these cells in the end of the nephron, so in these distal parts of the nephron. They're kind of touching these arterioles that are going into the nephron in the first place. So what they can do is they can sense the amount of fluid that is flowing past them, the amount of fluid that's about to get excreted, essentially, right? They can sense that amount of fluid. And if they sense that there's a lot of fluid going past them, right? They sense that there's tons of fluid that you're about to pee out, they can send a signal back to the arterioles going into the glomerulus. So basically, if you have a lot of fluid leaving the nephron that you're about to urinate, that probably means that there is a lot of fluid coming into the nephron in the first place. So that's a way in which it's kind of an indirect measure of the glomerular filtration rate. You know, it's not an exact measure because we have a bunch of reabsorption happening. But if you had runaway increases in blood pressure and therefore runaway increases in glomerular filtration rate, you'd also have runaway amounts of urination, essentially. So these cells in the ascending limb of the loop of Henle or in the distal tubule, they excrete paracrine factors, and all a paracrine factor is like, it's like a hormone but local. So it's a cell signaling molecule, but it doesn't go far, it doesn't go into your bloodstream, it's just going to these neighboring cells. So these macula densa cells are communicating with the walls of our arterioles. And then we have the same situation as with the myogenic regulation. So again, our arterioles are going to contract. And when they contract, the radius decreases, which means their resistance increases, which means we have less blood flow. So this also decreases our glomerular filtration rate. And then our final one is mesangial cell contraction. So this one is honestly less well understood. And when I was doing a little extra research on this to make your packets with your labeling diagrams, some of the sources say they don't believe mesangial cell contraction actually happens. But the theory is that the mesangial cells are able to actually sort of contract pieces of the glomerulus itself. So to make those capillaries tighter, which would also decrease GFR. So basically what I'm saying is all of these methods of regulation are mostly designed to decrease our glomerular filtration. So mostly we're seeing these in response to rises in blood pressure, or at least the way we've explained them here. These are all telling us that we're noting either through stretch of the arteriole or through fluid flowing through the nephron, we're noting that we've had an uptick in our glomerular filtration rate, probably due to a really high blood pressure. And we're trying to use this intrinsic regulation to bring it back down. And this is just a zoomed in picture of a histological site in case you were curious about what that looks like in reality. So this thing is our glomerulus. This is our capsule. And here's that end of our tubules. So probably the ascending limb of the loop of Henle, potentially our distal tubule. Okay. So here we see a little flow chart of ways in which we are controlling our glomerular filtration rate. So all we've done is divided the myogenic regulation. So that's the one with the muscle stretch, which is why we see myo, versus our tubuloglomerular feedback. So tubulo having to do with the tubes. That's the one about the flow through the distal parts of the nephron. So all that we're really going to see different here is why we're happening. So we'll note that these are both responses to increases in MAP, right? So increases in that mean arterial pressure. We're noticing, basically that we're going to be, what did I say? Did I say something backwards? Possibly. Okay. So we're increasing this. We have an increase in pressure in the afferent arteriole, which means we're stretching the arterial or smooth muscle, which means we are increasing constriction, which means we are increasing the resistance, which ultimately is decreasing the glomerular capillary pressure, which is a negative feedback loop. Oh, I see what's wrong. So it's a negative feedback loop. So the next effect is that it decreases the glomerular filtration rate. I see what they've done. Okay. And what this map is telling you on the left-hand side over here is that the direct effect of the increase in pressure in the arteriole without any regulation would be to increase pressure, right? To increase pressure and therefore to increase our glomerular filtration rate. And this is what we're trying to prevent, right? This is what we're trying to stop this. Myogenic regulation is a negative feedback loop. So myogenic regulation kind of turns this off. Regulation prevents. Okay. So the negative feedback loop is we're trying to turn off this system, basically. So that's the same thing happening on the right-hand side with the tubuloglomerular feedback. So we're still trying to do negative feedback here on the glomerular filtration rate. So we're still trying to turn off this direct effect of a rise in pressure. The only difference is we're doing it a different way. So in this case, what we see is that the glomerular filtration rate is going up initially. So we're noting that we have more fluid going into the nephron. But what happens is that means we notice that there's more flow going through the distal parts of the nephron. This is telling those macula densa cells to send a signal. So we have paracrine secretion. So that's the signal going to the arterioles, which ultimately constricts them. That's the same. Increases resistance. That's the same. Decreases the glomerular capillary pressure and therefore like turns off this loop. So that's how we again decrease our GFR. Okay. So I do feel these figures are a little bit confusing. So questions about them at this point? As you're looking over them when you're studying, if these become more confusing again, please do let me know and we can talk about them again. Okay. So those methods of regulation were how we're controlling the mean arterial pressure here in that plateau, basically. So this is really mostly where our intrinsic regulation is taking place. We also do have extrinsic control of glomerular filtration rate. And so when we think extrinsic, we're thinking nerves, and sometimes we're thinking of hormones from other parts of the body. Those are extrinsic factors. So extrinsic factors are just from somewhere else that's not the kidney. So our extrinsic control of glomerular filtration rate are basically ways that we're controlling blood pressure as well. So if we have a decrease in blood pressure, this can decrease our glomerular filtration rate because we have less blood flowing through our afferent arterial, so we have less blood in the glomerular capillaries, so we have less fluid pushing on those walls, which means our glomerular capillary hydrostatic pressure is lower. So this would tend to lower our glomerular filtration rate, which we can see in this section here. All right, so over here, we can say that if we don't fight against this, low blood pressure means low filtration. Okay. But we can control some of this through extrinsic controls. So our intrinsic controls were really about controlling for high blood pressure. We're going to use some extrinsic controls if we want to raise a low blood pressure, basically. So we can stop here and start with feedback loop on Friday. I think that's our best bet. But some of this should look real familiar, right? Our baroreceptors, our nerves, all those things that related to the heart and our blood pressure are things we've looked at before. So while this diagram looks a little scary, it's pretty much mostly stuff we've already discussed.